Okay, so today's lecture is going to be on meiosis. Not to be confused with mitosis, there's only one letter difference. This section is actually found in chapter 11, but we're going to be including it today with our chapter 10 discussion. Okay, and the whole purpose behind meiosis is we are making gametes, in other words, our sex cells. So let's take a look at our objectives. Okay, here's our list of our objectives. Make sure that you are able to answer these objectives in sort of a short answer or essay format at the end of the lecture to ensure that you have comprehended the material. Okay, so when life begins for all sexually reproducing organisms, we combine an egg and a sperm cell to produce our offspring. Okay? In humans, our egg and our sperm cells are going to contain a different number of chromosomes from the rest of our body cells. As you recall from mitosis, human body cells contain 46 chromosomes. But if we were to, here, let's add this. But if we had 46 here and we had 46 in the sperm, that would give us 92 chromosomes. Okay, then the offspring would have too many chromosomes, and that would result in a severe mutation. And actually, the development of the fetus would never occur if this were the case. Okay, so the way that we solve that is by cutting the number of chromosomes in half. So this is specifically just for sexual reproduction. We're going to describe the way that the cell divides so that it reduces the number of chromosomes in each cell by half. And that makes sense because then when we take that egg cell with 23 chromosomes and we add it to a sperm cell with 23 chromosomes, then the first cell of that offspring, which is a body cell, will have 46 chromosomes. Okay? The terminology that goes with this full set and half set of chromosomes are diploid and haploid. Okay, so most cells in the body are considered diploid. In other words, they are 2N, and that's just a formula that we use for them. Okay, so this would be like saying, humans are diploid with the number 46. Okay, that's because we have homologous chromosomes. We have 23 pairs. Remember, this one comes from mom, and let's say this one comes from dad, okay? And we're going to have 2 of each chromosome type. So 2 times 23 gives us 46. So we say all body cells or all somatic cells are going to be diploid. Well, when we need to cut that number in half, then we have to make some choices here. Instead of wanting 2 of every chromosome, one from mom, one from dad, we're going to pick and choose to have only 1 copy of each chromosome. This is known as the haploid number or the 1N number. Okay? And in sexually reproducing organisms, our sex cells, like our sperm and our egg, are the only haploid cells in our body. So every other cell in the human body contains 46 chromosomes, but your sperm or your egg cells, they only contain 23. Important here that you could describe the reason why that happens, why we only need 23 chromosomes in each of our sex cells. Very important part. Okay, so let's review mitosis. All right, mitosis, we produce 2 cells. They are genetically identical to that original parent cell and also to each other. Those cells are going to be diploid, and they're going to make somatic cells or body cells. The function of mitosis, which is down here, okay, functions down here, we're trying to grow, okay, or repair from an injury or replace worn out cells. So the idea is we're trying to make identical copies in mitosis. Meiosis, however, we're going to go through 2 rounds of division, so that's going to give us 4 cells. And because of the way that the dividing is going to happen, they will be genetically different from each other and also from the parent cell. This is going to make 1N or haploid cells, and the purpose is to form gametes for sexual reproduction. Okay, so what you should see forming between these two slides are some key points in a comparison and contrast essay, wink wink, nudge nudge, between mitosis and meiosis. So what are the reasons why meiosis is different? Okay, there are 3 things we want to highlight because it's not important to go through all the phases. You already know, in prophase, your nuclear membrane breaks down, and in anaphase, you're going to have the chromosomes separating and moving to opposite sides. That's not as important to focus on in meiosis. You already know what happens in the phases. Let's talk about what's going on with the genetic information that makes it so different from mitosis. First thing, remember those homologous chromosomes that we've talked about before? Those guys right there, okay? We color code them so that you remember one comes from mom, one comes from dad. They are not identical to each other. They are homologous. But what's going to happen is in the first division in prophase, so we call that prophase 1, that's not an I, that's a 1, they're going to group together. So they're going to find each other. Okay, this is called synapsis. Okay. And when we look at this group, this one right here, let's see. Okay, and when we look at this group of 4 chromatids together, it is known as a tetrad. So here you have, mom's chromosome number 5 plus the copy, and dad's chromosome number 5 plus its copy. These are going to group together and stay associated with each other. Okay? Sometimes when they're grouped that close to each other, they kind of get a little tangled up with each other. And if we see here, what has happened is something called crossing over. The arms of the chromatids have exchanged genetic information, as you can see here by this color coding. So now what's going to happen is, do we have sister chromatids that are identical to each other on the same pair? No, we do not, okay? This chromosome is still all of, mom's chromosome 5. But now this guy, this is mom's chromosome number 5, except down here at this piece, where now it has dad's chromosome number 5 piece on it, okay? This is going to allow for what we call the shuffling of genetic material, or it's going to help increase genetic diversity. Okay, so just reviewing again, homologous chromosomes, okay, they are not identical, okay, they have the same genes, and they're the same chromosome number, but they could be different expressions of those genes, right? One could be dominant, one could be recessive. So now when you add crossing over in, you're going to create new combinations between the maternal and paternal chromosomes. So this rearrangement of DNA, now when we look at an individual sister chromatid, they're no longer identical, right? Here on this side, we see that this chromosome arm is identical to this chromosome arm. But over here, crossing over has occurred between these 2 arms here, and because of that, they are no longer identical. So if we were to look at this and say, okay, well, if each one of these single chromatid arms are going into a separate cell, would any of those cells be the same to each other? And the answer is no, okay? Because this one would have, pink, dark pink, and green as its chromosome combination. This one would have blue, dark green, and green as its chromosome combination. This one would be pink, red, and blue for its chromosome combination. And this one would be blue, green, and blue for its combination. So we have created 4 different combinations. And again, the key purpose of that is to increase genetic diversity. So is it a mutation? Yes, I mean technically, but mutations aren't necessarily bad things. And in this case, for sexually reproducing organisms, we consider crossing over to be an advantage. Creating more genetic diversity means we're more likely to be able to adapt to our environment should there be something, bad out there. Okay, there are a lot of varieties of genetic combinations, and somebody's going to be able to survive and overcome the environmental problem. All right, so second thing that makes meiosis different, when we separate in anaphase 1, so 1 meaning the first round of division, what's going to happen is there's nothing controlling the order or the way in which these chromosomes or these homologous pairs line up. Let me show you a diagram. Okay, so here we see a parent cell, okay, and because of segregation and crossing over, we can make all these different combinations, as I said before, right? So there are your 4 separate cells. How does independent assortment play a role in this? Let me show you. Okay. When your tetrads line up in the middle of metaphase, there are 2 options for how they could line up. You could get both paternal chromosomes on the same side, or you could have them crisscross from each other. Okay, so when we go through division and we ultimately separate, this possibility, so cover up 1/2 of your screen right now. Let's just look at the left side. Okay, over here the possibilities are that they both come out with maternal or both come out with paternal. Okay, whereas in this arrangement they get 1 of each. Okay, and this isn't even looking at crossing over, so we need to put that aside for right now. We're just looking at the movement of the chromosomes and what that's going to allow for. So there's nothing controlling whether the paternal chromosome winds up on the left or whether it winds up on the right, as we see here. So both of these arrangements at metaphase are completely possible. So we're saying that these chromosomes, okay, this tetrad and this tetrad, they're going to assort independently of any other control. So it's really sort of a flipping to chance. So that adds even more genetic diversity into the mix. Okay? Look at all these different possible combinations. And again, we haven't even taken into account the additional genetic diversity from crossing over. So this is why you don't look exactly like your siblings, even though your parents' gametes came together each time. The gamete and the gamete that they happen to donate in that union could be completely genetically different. Okay? So this is just a quick little animation to show the process of meiosis. We're going to go through 2 divisions. So here we see tetrads forming and crossing over occurring. The end of meiosis 1, we have 2 cells. Let me show you this. Okay, so this is meiosis 1, pro-meta-anatel. There we go. And here's meiosis 2. So at the end, we have 4 genetically different cells, and they are each containing half of the original genetic information. Okay. The last thing that makes meiosis significantly different is the fact that we skip interphase when we go through the cell cycle. So if we look at this diagram down here, let me get a pen. Okay, if we look at this diagram down here, we have interphase, we have prophase, metaphase 1, anaphase 1, telophase 1. Okay? So right here, this is the dividing line between the first division and the second division. Okay? Now, if we were doing 2 full cycles of the cell cycle, at the end of the division, we would have gone right back into interphase. But it's important. There is no... Oh, I'm sorry. Let me try that again. This pen. Okay, but it's, here, I'll put this line here, and it's important to say no interphase. Okay. So what's going to happen is we get two cells and they're immediately going to start going through division again. So we jump right from telophase 1 into prophase 2. Except now it's happening to two cells at the same time until our end result here we have 4 haploid cells. Okay. If we did allow interphase to occur, what would be the issue? Well, we would never be able to cut our chromosome number in half because we'd constantly be doubling and dividing and doubling and dividing and doubling and dividing. And each time we double and then divide, that would cancel out any chromosome number change. So if we're talking about humans, let's say during interphase, we started with 46, then we go to 92, then we divide, and each of these cells have 46. Okay? If interphase were to occur at this point, we double back up to 92. That doesn't work. But if we keep it at 46 and then go through division, each of these cells now have 23. Okay. Here's a little schematic to help you compare mitosis versus meiosis. Okay, specifically again talking about a human cell. We start with 46. Then during the S phase of interphase, we're going to double it to 92. And then at the end of mitosis, we split 46, and these cells are identical to each other. Okay. Whereas with meiosis, 46 to 92, yep, this is still interphase. Okay. And then from here to here, this is the equivalent of meiosis 1. So that is your first round of division. But as soon as we finish with meiosis 1, we enter the second division again. And so this would be meiosis 2. And the end result is that we cut our chromosome numbers in half from our starting number, and they are all genetically different. Okay? Okay, so let's talk a little bit about how sperm and egg formation actually occurs. We know that meiosis is going to take one diploid cell and divide it to form haploid cells, but male and female gametogenesis are actually separate from each other. Okay, spermatogenesis, this is the process of making mature sperm. Okay. The sperm cell, which is located here on the left, very, very small compared to the egg or the ovum. Okay, better terminology. Inside the head is where the majority of the cell body is, and there's barely enough room for the nucleus, so it just contains the chromosomes. The only other thing it needs is obviously going to be a flagellum, because the sperm needs to be mobile or motile in order to swim its way to the egg cell. So if the sperm is only providing the DNA, then that means everything else of the egg, the nutrients, the organelles, and things like that, come from the egg cell. Okay. This is my favorite word in biology just because I like how it sounds. Oogenesis is the making of a mature egg. Now, things are a little bit different here. With a sperm cell, we have one starting cell and it divides to form 4 sperm cells. But eggs, they're a little more about the quality. And what's going to happen here is we go through our division, right? This represents meiosis 1. But look at the size difference between this cell and this cell. Okay, it looks as though they divided unequally. And so all of the nutrients and cytoplasm and good stuff went into this cell, whereas this cell is really non-existent. Okay, then both cells still go through the next round of division, so meiosis 2 occurs. And when we end, we have, again, an uneven division of the cytoplasm and nutrients. So finishing up, we're going to have what are called polar bodies. And what's going to happen here is when we look at each round of meiosis, you get 4 sperm cells from each round of meiosis, but you would only get 1 true egg cell from each round of meiosis. These other guys, these polar bodies here, they're going to die. Okay? And let me explain why on the next page. Okay? Why make only 1 good egg? Well, it's a quantity versus quality thing. When you think about the development of the offspring, it's generally going to happen within or due to the investment of the female. So the female is really about quality. And so when the female is forming the egg, the female wants to make one really good, nutrient dense, energy efficient egg cell that's going to be like presenting the best of herself to this offspring. The sperm, little less involved in the quality and more about the quantity. I mean, honestly, the whole idea of spreading your seed, okay, in many different species, the males do not play a big role in raising young. Instead, they are just, concerned with getting as many females to produce their offspring as possible. But since the female is the one who is carrying the offspring or caring for them, she wants to make sure that she gives everything she can to that offspring. So that's one of the reasons why one good egg comes out of that, and those other 3, those polar bodies, never develop into egg cells. Okay, what happens to them is they're going to self-digest. So we're going to use the lysosomes, and we've talked about this before, but just again here, that is going to be apoptosis. Okay, what's going to happen is those polar bodies are going to go into this degeneration or cell suicide program, and they're going to be broken down and destroyed. But it's okay because we got that one really great egg cell out of it. Okay, make sure that you go back, check your objectives. Again, key focus here is being able to compare mitosis and meiosis. If I had to pick the one objective that you should really know, sounds kind of essay worthy, right? A compare and contrast essay, mitosis and meiosis. Make sure you know it.